sample. Next is when we talk about DNA or RNA sequencing. So there is a common approach that first we extract the samples, then we get the fragmented DNA, attach the adapters, and then provide to sequencers. So first step will be input your DNA, second fragment it, third is add the adapters, and then provide it to sequencers. So after the sequencing, what we need to do after sequencing, we have to align our reads or the samples on the reference genome, the sequences which are available on the DNA that are already being sequenced, we call it reads in the general term. So when we match that diseased read to our reference genome, that is the normal read, to the exon or the protein coding region in case of RNA, so, or transcriptomics. So we try to match, and what is the section of the read of your sample is matching to the reference genome. So that is what we call the alignment or mapping. So whenever we match, so it is not necessary that every time the read that you get in your sample is matching exactly to the reference genome. So sometimes it will half match, or less than half it will map to the reference genome. So that will be called unmapped reads, and that may be one of the cause for the disease. So we need to further explore that difference, that why it is not mapping to the normal genome, which is a reference genome. Next is the industry uses. So there are different type of industries where the NGS is being used, even it is the biology specific or the technology specific. So in biology specific, you will find different insights that why this gene is differently expressing, what pathways it is involved in, and what is its expression in that sample. Next is technology industries, where these sequencers or other tools to process the samples are being developed, so that the sequences that we are retrieving from the patient, that can be sequenced. So based on our interest, we can see that what type of industry we are interested in. Either we are interested in like research and development, we are interested in technologies, or even computation, where new software you can develop, you work with the new softwares to identify or to find the biological insights of your data. Next is, so based on different industries, we have academic research, clinical research, hospital applicability, biopharma, biotech. There we can work in forensics or cancer screening, epidemiology, drug development. So based on our interest, we can see that what type of industries are over there and where we can work. Next is career opportunities. So while choosing a career, we first need to talk to ourselves that what we are interested in. So there are a couple of questions that we can ask to ourselves, that do we enjoy making difference in people's life? Like, do we really feel for people, like people with the disease or some kind of shortcomings that can be treated? So are we really interested in it? Do we feel sympathetic, so that we can work more efficiently if we feel pain of others? Next is, do we like solving problems and challenges? Because once we are getting into this field, so we will have too many challenges to come over. So we need to ask these type of questions to ourselves, that what really we enjoy. Do we feel happy like working with the problems, solving problems, understanding problems? Next is, do we feel happy working with as a part of a team, like in a team, or you like working independently? So based on these questions, we will able to identify that where our interest lies in, and to which what type of jobs or industry we can apply and we can work with.